Aseptic techniques are performed in microbiology and general science labs all around the world. They help keep scientists, researchers, interns, and technicians healthy and safe, and they prevent contamination of experiments, which can lead to faulty results. In this video, we're going to talk about aseptic techniques, and we'll look at some common ones that are used in the microbiology lab. By the end of this video, you should be able to describe the term aseptic technique and give some examples. You should also be able to describe how to aseptically transfer bacteria between broths, and you should be able to explain how to sterilize inoculating equipment. Finally, you should be able to explain the proper way to remove and replace test tube and bottle caps. Aseptic techniques are the set of steps and protocols that we use in the laboratory to prevent contamination of ourselves, our experiments, and our environment with unwanted microorganisms. These protocols begin pretty much when you come in the lab and you put your book bag and your coat away, either in a cubbyhole or by hanging them on the wall. A clean and uncluttered work surface is essential to a good aseptic technique. So we want to make sure that all of our materials are basically put away and we have a clean work surface to do our experiments on. After you've put your materials away, you'll want to wash your hands thoroughly to remove as many microorganisms as you can from your hands. Be sure to use soap. This reduces the number of unwanted microbes brought into the lab from outside. Next, you're going to wash your desktop to remove microbes that may have settled onto this desktop from the air, or maybe they were left behind by a careless experimenter. I prefer using 70% ethanol or 70% isopropyl alcohol, but some labs will use detergents like Backdown instead. Whichever chemical you use, it's most important to pour some onto the desktop surface. Use a paper towel to move it around, but don't use the paper towel to dry the desktop. The amount of time that the chemical has on the desktop is actually going to increase how many microbes get killed. So we want to let these chemicals evaporate rather than wiping them away so quickly. After you've washed your desktop, you'll want to wash your hands again. You'll scrub them hard to get rid of any microbes that you might have acquired from the desk, and also to see if you can remove any last stragglers that you might have brought in from outside. After all of that washing, you'll apply the PPE, which provides a barrier between you and your experiments, so that microbes on your body and on your clothing don't contaminate your experiments, and so that microbes in your experiments don't splash up onto your body or clothes. Be certain that if you're wearing goggles in the lab, which you should be, the goggles have closed sides like these, so that splashes that might occur to the sides of your body still won't reach your eyes. You also want to make sure that your goggles are ANSI Z87.1 rated. This means that the goggles will protect against impacts, UV light, and chemical injury. Now, another precaution to take is with your gloves. When you put your gloves on, you want to make sure that the cuff of your gloves goes over the cuff of your jacket. This makes kind of like a seal that's going to give you added protection to keep microbes from getting onto your body through your PPE. One of the first labs that we do in microbiology is the subculturing lab. Subculturing is a routine technique in which microorganisms, in our case bacteria, are transferred from a culture to sterile media. So that means we're going to have a turbid broth or a petri dish that has colonies on it, and we're going to transfer those microbes into a sterile medium. Why would we do this? Well, we need to keep microbes growing in the lab so that we can do future experiments. We might be doing this transfer in order to generate pure cultures of bacteria. Maybe we want to grow the microbes just to store them for later or to maintain the cultures. All of these are valid reasons to subculture. The first step in any microbiology experiment is to label your materials. If you're working with petri dishes like this one on the left, you're going to put a label on it. If you're going to work with test tubes like what you see on the right, you're going to put a label on that as well. Pause your video for a minute and write down some of the things, some of the information that you think belongs on labels for these pieces of equipment. Once you have your list, join me again and let's compare notes. So one of the first things you'll want to have on your label is the name of the microorganism that you're working with, or whatever your specimen name is. The second thing is the growth temperature for your specimen. Where are you going to incubate it? At 37 degrees Celsius, room temperature, which is usually around 20 degrees Celsius, or is it going to be 30? That should be on your label. Next, you absolutely should put the date that you're doing your experiment or your subculture on the tube. That way, in a couple of weeks, when you go back to see how old the bacteria are, you'll know. The fourth thing that belongs on your label are your initials. This way, everyone in the laboratory knows whose tubes belong to whom, and people don't accidentally do experiments with other people's resources. Finally, you'll want to label your tubes and your plates with what medium is in those tubes and on those plates. A lot of microbiology media look the same. They're all kind of goldish colored liquids and augars, or maybe they're all pink. It can be hard to distinguish them, so writing the name of the medium on the actual tube or petri dish is also important. Now, when you're putting your label on a petri dish, you want to label around the outside of the dish. And the reason for this is the microorganisms are going to mostly be in the middle. So we don't want to obscure them with black lettering from a Sharpie marker where our label happens to be. So instead, I recommend writing on the outside edge where there's going to be fewer microorganisms and you can still label the plate. You always label the bottom of the petri dish. The bottom of the petri dish is the side that has the agar in it. We never label the top because if the petri dishes get jostled in the incubator or if they fall to the floor and the labels for the dishes are all in the caps will come off of the dishes and roll across the floor, and now we don't know what's on the agar. So you always label the side of the dish that has the agar in it. As far as test tubes, I recommend labeling in the middle of the tube. You want to stay away from the mouth of the tube up here where the cap is, because the chances of you putting bacteria onto the tube are pretty good, and if you put those bacteria around the mouth of the tube, there's a good chance that you'll contaminate your bacteria. The distance between the contaminating bacterium and the inside of the tube is very short if you label near the mouth of the tube. You also don't want to label too far down at the butt of the tube because that's where your microbes are going to be growing, and we want to be able to observe them to see if there's any turbidity, if there's any coloration, we want to be able to characterize anything that's growing, any growths in the tube. So we try not to label really low in the tube either. So that's why I recommend the middle. The same information, the name, the growth temperature, date, initials, and medium would go on your test tube or on the agar plate. Now, let's watch a video on Aseptic Technique by Biorad together. The demonstrator in this video is subculturing bacteria, and it's going to do a number of different subcultures.
In this video, you will learn the proper practices and procedures for performing aseptic technique. Using aseptic technique, you will be able to transfer a bacterial monoculture into a fresh medium without introducing any contaminating microorganisms. Two of the most commonly used tools for transferring bacteria are the inoculation loop and the inoculation needle. Knowing how to use a Bunsen burner is an important skill in aseptic technique. To light a Bunsen burner, start by closing the valve at the base by rotating the collar so that the vents are covered. Then slowly turn on the gas. Once you hear the gas, ignite the burner and set the height of the flame by adjusting the amount of gas. The temperature of the flame can be adjusted by rotating the collar to control the flow of oxygen. The coolest flame is yellow and orange. A medium flame is blue. And the hottest flame is a roaring blue flame characterized by a clear blue cone in the middle. The tip of the cone is the hottest part of the flame. Inoculation tools are sterilized by passing them through the hottest part of the flame. Once sterilized, inoculation tools should not be set down on any surface.
Notice that as he sterilized the inoculating loop, he allowed each part of the metal on the loop to glow kind of an orangish yellow, maybe a reddish orange. That is essential and very important. If you don't allow the loop to glow like that, you won't kill any bacteria or all bacteria that might be on the loop. There are some bacterial cells called endospores that are extremely heat resistant, and it's only when you get temperatures that are hot enough to turn the loop that yellowish orange that you can kill those endospores. The other thing I want to mention is, just as the experimenter said, or the demonstrator said, you don't want to cool your loop by touching any other surface. So a few years ago when I was teaching this lab, there was a student who was trying to cool her loop by touching it to paper towels. And I've seen other students try to cool their loop by touching it to the back of their glove. The problem with that is you're completely negating all of the effort that you did to sterilize the loop. You can pick up bacteria from the paper towels as well as from the back of your hand. They go onto the loop, and then when you use that loop in your experiments, you'll contaminate them. So, never let the loop touch any other surface except for the surfaces that have the bacteria that you're interested in working with.
In this section, you will learn how to transfer bacteria from a liquid culture to a petri dish. After flaming a loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip.
Really quickly, why do you think the demonstrator is holding the cap from this bottle in between his ring and pinky finger? Right. If he places the cap onto the desktop, he runs the risk of bacteria from the desktop getting onto the cap. Then when he puts the cap onto the bottle, some of those bacteria may wind up inside the bottle and contaminate the culture. Notice that he's holding the cap so that the open side is facing downwards. That's to keep microbes that might be in the air from landing in the cap, so that, again, once he puts the cap onto the bottle, those microbes won't fall in.
Dip the loop into the liquid culture and flame the mouth again before replacing the lid. Transfer the bacteria to your petri plate. Lift the lid of the plate and hold it over the base as a shield to prevent contaminants from falling onto the plate. Spread the bacteria over the entire surface of the plate in all directions. Rotating the plate a quarter turn between spreads will ensure that the bacteria is spread evenly across the plate. In this section, you will learn how to transfer bacteria from a liquid culture to a liquid. After sterilizing an inoculation loop, take the lid off the container of liquid culture and pass the mouth through the flame. Then dip the loop into the liquid culture and flame the mouth again before replacing the lid.
Why do you think it's important to flame the mouth of the bottle? Right. In the off chance that bacteria were deposited onto the mouth of the bottle either during this manipulation or a previous one, we want to kill those because they may not be desired microbes. They could be contaminants. So passing the mouth of the bottle in this case through the through the Bunsen burner flame should kill any microbes that might have landed on the surface of the bottle mouth, either on the outside or on the inside.
Now take the tube of liquid media and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Flame the mouth of the tube before replacing the lid. Flame the loop to sterilize.
Why is it important to flame the mouth of the test tube after you've done the transfer? Right. It's the same reason that we were flaming the mouth before, is to kill any microbes that might have inadvertently landed on the mouth of the test tube. Remember, the goal of aseptic technique is to prevent contamination with unwanted microbes. So each of these flamings between the putting the caps on and taking them off, all of it is to prevent contamination. Oftentimes, students will forget to do the last flame after they've done their transfer. I hope that you won't forget that. One more thing. Why do you think it's important to flame the inoculating loop after doing the transfer? Right, if we're going to lay the loop anywhere, like in a drawer or on a table, and we don't flame, then the microbes from our experiment will contaminate the surface where we've laid the loop. The only way to kill the microbes that we've been working with is to pass them through the flame of the Bunsen burner and sterilize the loop. This is an important part of aseptic technique, and it helps prevent contamination of our environment.
In this section, you will learn how to transfer bacteria from a petri plate to a stab culture. Start by sterilizing an inoculation needle by passing it through a flame. Allow the needle to cool. You can test for coolness by tapping the needle in a clear area on your agar plate. While holding the lid as a shield, gently rub the inoculation needle over a single colony. Transfer the bacteria to a culture tube filled with nutrient agar. Remove the top of the tube and insert the needle, making 3 deep stabs. When finished, replace the lid on the tube and sterilize the needle. In this section, you will learn how to transfer bacteria from a petri plate to a liquid. Sterilize an inoculation loop by passing it through a flame. Let the loop cool. You can test for coolness by tapping the loop in a clear area on your auger plate. While holding the lid as a shield, gently rub the inoculation loop over a single colony. Now take the tube of liquid media, remove the lid, and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Pass the mouth of the container through the flame before replacing the lid. Sterilize the inoculation loop to complete the process.
Okay, let's review. How would you subculture bacteria from a culture to a broth? Well, before you start the experiment, you've got to label your culture media. In this case, we'll have two test tubes. So you'll want to make your label in the middle of the test tube, preferably with a Sharpie marker that you write directly onto the tube with. Next, you're going to light your Bunsen burner and adjust it so that you have a dark blue outer cone out here and a light blue inner cone here. Remember, the interface between the two cones is the hottest part of the flame, so you'll want to insert your inoculating needle into that region. You'll also want to hold the different parts of the wire for your inoculating needle in the flame long enough so that the wire glows kind of an orangish yellow color. Next, after you've flamed your loop, you're going to remove the cap from your culture tube. So you'll do this by holding the cap of the tube in your same hand where you're holding the inoculating loop. This is actually the way that I hold the cap in this image versus what you saw in the Biorad picture. I just feel like the cap is more secure. However you hold the cap in that hand, you want to make sure that the mouth of the cap is facing downwards so that microbes don't fall in, and then you run the risk of contaminating your experiment with those microbes. After you've removed the cap, you're going to want to flame the mouth of that test tube. Next, you'll use your sterile inoculating loop, which by now has cooled, to remove your inoculum. You're going to stick the inoculating loop into the tube and bring it out, trying not to touch the sides of the tube. Then you're going to flame the mouth of your test tube, that's the one with the culture, and you'll replace the cap. After you've replaced the cap on that tube, you'll set it aside and pick up the test tube that has your sterile broth. Notice that you pick up the test tube with the hand that's opposite the one that has the loop in it. So, not the hand with the loop. You're going to use the hand with the loop to remove the cap from the test tube, as you see here. Then you'll flame the mouth of the test tube and insert your inoculum. After you've inserted your inoculum, you're going to remove the loop, flame the mouth of the test tube again, and then cap the tube. Are you done? No, we've got to flame the loop. That's the last step of our aseptic technique. We want to be sure that we don't contaminate the environment around us with bacteria from our experiment. So that brings us to the end of this video. In this video, I've described aseptic technique as being the set of steps and protocols that we do to help prevent contamination of ourselves, our experiments, and our environment with unwanted microorganisms. We said that technique begins when you enter the microbiology lab and you put your materials away. It continues when you wash your hands to get rid of any extra microbes that you might have brought in the lab with you, and then when you wash your desk. Remember, after that, you wash your hands and apply PPE, all of which is part of aseptic technique. You know what else is part of aseptic technique? Flaming your inoculating loop and flaming the mouth of your test tubes. We said that flaming sterilizes inoculating equipment like loops and needles because we're going to raise the temperature of the metal hot enough to kill all forms of microbial life. We also said that another way that we observe aseptic technique is to hold the caps to our tubes and bottles using our ring and pinky finger of the same hand that we're holding the inoculating loop. This way we don't have to put those caps onto the tabletop where they might pick up more microorganisms. I know this was a lot of steps, and this may not have been the most exciting video, but hopefully you feel somewhat confident that if you had to do this in a laboratory, you could accomplish an aseptic transfer with minimal instruction. That's my goal. Now, what I'd like for you to do is take a look in your course space for another video. We're going to watch this one together, and this time we're going to look for lapses in aseptic technique. I look forward to seeing you there.